First of all, I thank Dr. Lee for giving me this opportunity to be in a clinical meeting. I think I'm seeing very different topics and data sets. Maybe my talk will take a different turn for your mind. My lab is very much interested in the fundamental aspect of synaptic development and neurotransmission. I think recently we have developed some interest to study autism. So let me give you a quick overview on what are we talking about. So one of the emerging themes in autism research is that cerebellum is now being considered as a major mediator of autism risk. This is a big data analysis. As you can see, that cerebellum injury at birth actually is among the most important autism risk factor. And premature baby, for example, also is one of the relevant factor. But surprisingly, if you consider this, genetic underpinning of autism is not a major cause of autism. Rather, cerebellum injury, something about cerebellum in the early development brain that primes the manifestation of autism. And this is probably one of the very important risk factor that isn't being very well recognized. The reason is that we know cerebellum is an ancient brain. It only probably is important for motor social functions. And it turns out that this small area of the brain, it takes about 7 to 10% of brain volume, but contain 70% of neuron of the entire brain. I think most of you may actually consider neocortex as the main loci of psychiatric disorders. Surprisingly, cerebellum now is emerging as a key factor priming many different neuropsychiatric disorders. The reason is that early cerebellum connection through thalamus to cortex, then come back to the cerebellum. This cerebellothalamic cortical loops appear to be play an important role in priming the brain development. And so the circuit is quite simple. In fact, it's very modular. So let me just move on quickly. So the granule cells, as I mentioned, are numerous, and they receive all kinds of sensory inputs from your visual system, olfactory, and vestibular system, whatever. They actually send projection into the cerebellum cortex to contact the Purkinje neurons. And the Purkinje neurons, the only way for entire cerebellum the only gateway for the output of entire cerebellum. So excitation through MOSI fibers that integrate all sensory informations, and the climbing fibers are excitatory inputs that make correction for motor learning, for example, are the inputs, and they are controlled by inhibitory interneurons in the cerebellum to gate this EI balance, how much output is generated from the cerebellum. So Purkinje cell is very vulnerable to damage or injury at birth, for example. One of the most important genetic evidence to suggest cerebellum play an indispensable role in the pathogenesis of autism is this study by Albert, and this was the review in Nature in 2013. This paper, Peter Tsai published in Nature, shows that a single gene knockout from Purkinje cell, this is TSC in the mTOR pathway, that can lead to system-wide autism. If you consider you have Purkinje cell only and one gene knockout leading to system-wide autism, this argues for the critical role of autism for the pathogenesis of cerebellum in the manifestation of all behavior phenotypes. Now there are 5-6 cell-specific knockout genes now, SHANK3 and P10 and GRIP1, 2, and all of this showed that convergent phenotype, that Purkinje cell firing substantially reduced. And so we got into this using FMRP mice as a model, because we know Fragile X syndrome is a leading genetic form autism with deletion or truncation of a single protein known as the Fragile X mental retardation protein. This is an mRNA binding protein, and what it does in the neuron is sequester mRNA into phase-separated granules upon stimulation, for example, G protein coupled receptors at a phase separate and translate numerous synaptic proteins that regulates neurotransmission. And in fact, we recently published a paper showing the minimal structure for this phase-separated granules residing in the C-terminals. However, the N-terminal function of FMRP now is considered as non-canonic and very important for autism. And here is the data we made a recording from Purkinje neurons, and we find that indeed in the FMRP knockout mouse, they fire less than the wild-type control. But if you block excitatory inputs, you don't see much difference. But if you block inhibitory synapse, you find that a tremendous increase in the firing rate and very much comparable to the wild type. So if we consider this model, we know the inhibitory inputs onto the Purkinje cell by basket cell or stellate cell suppress the firing, and blocking the inhibitory synapse seem to normalize the phenotype. That suggests the inhibitory overtone that tip over the excitation inhibitory balance. And indeed, when we make recordings from the Purkinje cell, we can see they have excessive inhibitory synaptic currents. But if we block action potential, they get the synaptic unitary synaptic currents are no different, suggesting the nerve terminal of interneuron are highly excitable, and they generate numerous action potential and produce this inhibitory overtone in the cerebellum. Therefore, that gates the phenotype of inhibitory, gates the phenotype, a loss of function of Purkinje cell in its output. And this phenomenon we have seen before. Many years ago, we started using forward genetics, we discovered Kv1.2 channel OPSI that generate exactly what we saw in the FMRP knockout. So we began to realize maybe the Kv1.2 channels on the nerve terminal are downregulated. That's why you fire excessive action potential, drive GABA release, therefore inhibitory overtone. And we look at the Kv1.2 labeling in the cerebellum, we can see the basket cell nerve terminal that hugs Purkinje cell body express much lower level of Kv1.2 channel. You can see zoom in picture, Purkinje cells are here, the basket cell on the soma, that the pincer-like structure are sort of shrinked in the knockout mice. Both protein and mRNA level of Kv1.2 are substantially reduced. So to make a long story short, we eventually figure out actually the N-terminal I was talking about of FMRP produce this protein-protein interaction with Kv1.2 channel, C-terminal. And this interaction boosts the gating of Kv1.2 channel, therefore is an important drug target for the autism phenotype. And indeed, we looked many drugs. We discovered one of the fatty acid lipid, omega-3 fatty acid, DHA, can function as an allosteric positive modulator Kv1.2 channel and then normalize synaptic deficit. This is the experiment we show that DHA shift the activation curve of Kv1.2 to the left. And in this case, acute application to the slice where in the FMRP knockout, we have lots of inhibitory inputs onto the Purkinje cell can be suppressed by DHA at a very low concentration. And there's a firing phenotype that we saw typically in the FMRP knockout can be normalized by DHA treatment. So somehow this lipid can produce a very positive effect to normalize synaptic phenotype. And then at the behavior level, one injection of DHA suppress the startle response quite substantially. This is one of the best known phenotype in the FMRP knockout mice where the startle response were very large, and DHA seemed to be very effective in suppress. So our working model is that we believe FMRP produce direct interaction with Kv1.2, either to traffic, to the channel, to the nerve terminal, or from the soma to the nerve terminal, whatever that effect is, that allow the Kv1.2 channels to suppress the excitability of inhibitory neuron. Therefore, that the Purkinje cell can have a quite fast firing rate. A loss of the Kv1.2 lead to overwhelming amount of GABA release. And the DHA can function as the allosteric positive modulator, boost the function of Kv1.2. Despite fewer channels, they were able to reverse the behavior phenotype. So with all of this, so this work is published in the Molecular Psychiatry a couple of years ago. And so we followed up this work with brief DHA treatment as a dietary supplement. So we custom-made DHA into the rodent chew, and so the paradigm is that the early postnatal mice that we feed the animal with DHA plus diet or DHA sort of depleted or polyunsaturated fatty acid depleted diet compared to the naive. And then only 2 weeks that we treat the animal. Then we wait for animal to clear all the system in the system, DHA in the system. Then we perform behavior and electrophysiology analysis. So those are the elevated plus MACE is an indication of anxiety. Marble bearing is a repetitive behavior, an open field test, and pre-pause. So we have a lot of behavior tests, and then I just use one example here. We can see that in the knockout mice, they tend to have repetitive behavior. Now is completely normalized by DHA. Really a brief treatment in early developing brain can produce a long-lasting effect. And when we look at the Purkinje cell where we record iPSCs, this is again the very sort of extensive firing iPSCs onto the Purkinje cells. In the DHA-treated animal, this phenomenon is completely normalized. And what's remarkable is that when we look at the basket cell nerve terminal, in the wild-type mice, it has also a neurotrophic effect, promote the growth of inhibitory nerve terminal. But in the knockout, this effect normalized to a control level. So typically in the knockout, they have a small nerve terminal. Now they are actually completely normalized to a wild type level without treatment of DHA. And some of the behavior can be mapped onto the molecular markers. For example, we look at Kv1.2. Here is the open time in the arm, in the elevated plus maze. You can see that there is a strong correlation between Kv1.2 level and the time they spend in the open arm. And inversely, they are correlated with other molecular markers. For example, neuroinflammation markers, they're inversely correlated and positively related to the PSD95. PSD95 is a molecular marker known to be enriched in the postsynaptic neuron. Remarkably, actually, this marker also expressed in the nerve terminal. And this is one of the most important molecule known to interact with Kv1.2. And we performed at a cell level to FRET, which is a way to look at protein-protein interaction between Kv1.2 and PSD95. We can see that DHA treatment somehow promote the membrane association of PSD95 and Kv1.2. This is reflected by the reduction in the FRET time. This is fluorescence residence time when we use this technology to look at Kv1.2 and PSD95 phase separation. And so all of this suggesting that Kv1.2 can be stabilized by DHA even in the absence of FMRP. And another experiment we showed that is that if we perturb Kv1 interaction with PSD95, this which is very much described many years ago by Morgan Shin group, and we use this peptide, we can actually convert a wild-type inhibitory synapse to very much like a FMRP knockout if we use the peptide to perturb this interaction. This is reflected by a significant increase in the excitability of presynaptic GABAergic neurons, and they produce lots of inhibitions. So all of this lead us to conclude that a brief treatment with the unsaturated fatty acid can somehow magically reverse the phenotype of autism in the mouse model. And this have something to do with free separation of Kv1.2 and PSD95. In normal condition, we have FMRP, they interact with Kv1.2 to boost the gating and boost the trafficking. But in the absence of FMRP, the Kv1.2 channels are substantially reduced in the inhibitory nerve terminal. DHA, at this point, we are still working on the molecular mechanism, how DHA, this lipid, help phase separation that stabilize PSD95 and Kv1.2 association in the nerve terminal. But the big message, if you don't understand any of this, is that early brief intervention with unsaturated polyfatty acids have a very good rescue effect on morphofunctional and behavior deficit in the autism model for Fragile X. DHA facilitated recruitment and retention Kv1.2 to the inhibitory nerve terminal to normalize the behavior phenotype in the cerebellum. And DHA also produced an acute effect on Kv1.2 gating and long-term effect on synaptic structure via other signaling pathway that I don't have time to talk about. So now we actually, you take advantage of the structure, biology information of Kv1.2, determined that the DHA binds to the deep pocket in the Kv1.2 structure. Using this, we can, with the 3D structure as a template, we have been discover new compounds bind to this site, function just like DHA. And we have also discovered other compounds in different component of KV1.2 channels. So all of this can allow us to develop new drugs to treat a loss of function. I mentioned the study we did many years ago, but KV1.2 channels are associated with seizure, epilepsy, intellectual disability, and other developmental disorders and behavior deficit. But we believe that the cerebellum dysfunction, particularly in the developing brain, is critical to interfere early to produce the best therapeutic effect. I'll acknowledge the people who did the work. The first part of work is very much done by Amy Yang, now a tenured professor at the University of Minnesota, and Adam Facchetti and Jason Azenard. And Jason is still in the lab now, his work for the second part, the lipid supplement, is rescuing FMRP. And the KV1 mutant mice work were done by John Henriksen, and other work I don't have time to talk about. But thank you for your attention. This is my story.